So thank you, Martin, for the introduction, and good afternoon, everyone. So my name is Kieu, and I'm working from the Medical Center of Groningen. And today, in today's conference, I would like to bring you two stories. From the vascularization, I would like to emphasize another perspective from how the vascularization can be impacted during chronic infection or different cytokines that exposure to endothelial cells. And from another perspective, I would like to highlight our personal journey of how immunogeneticists like us can actually collaborate with engineers to bring up the blood vessel model on chip and use it to study endothelial cells memory. So firstly, I would like to invite you to travel back with me around 700 million years ago, far ago, and with the emergence of invertebrate organisms, and then around 450 million years ago, we also have vertebrate organisms. So during the long time span, I think we are all convinced about the importance of the immune responses and how critical it is for the survival of organisms. But maybe we forget that this function in the vertebrate organism is mostly guarded by what we so-called innate immune cells, including monocyte, neutrophil, and NK. And then lately, in vertebrate organism, we have another different class of cell type that are emerged that we call adaptive immune cells, that are B cell and T cells. And with T cell and B cells, adaptive immune cells are exclusively known for their immunological memory. So we know that we can use them for CAR T cell therapy, for vaccine therapy, but we don't know about that for innate immune cells. And recently, in 2015, a lot of publications have been collectively published to really show that these innate immune cells can actually also have memory. And this memory can actually fight to the better or the broader spectrum of infectious pathogens. So if you look at on the right scheme where we can see kind of like the map of how the trained monocyte would behave. So in the x-axis, we can see the time or the history of exposure to pathogen, and on the y-axis is the strength of the immune responses. So we can see that if the cells are infected for the first time, it can mount to a certain degree of infection or of immune responses. And then after that, it goes back to the homeostasis state, in which during this resting state, the cell can actually reprogram the metabolism, can modify how the chromatin impact or the epigenetics of the cells, and then ready for the second infection. So when it's encountered to the second infection, these monocytes can actually mount the much stronger immune responses. And in monocyte, we know that they secrete a lot of TNF-alpha in interferons. So this actually opened a really exciting period for us to really see how we can approach this trained immunity into disease therapy. But before that, we need to see what is the specific role of this trained immunity into the disease pathogenesis. So if you can see in the scheme here, indicate the strength of trained immunity. So a lot of papers have showed association of how strong the immune response it is and how associated that with different kind of diseases. So on the left side, on the red part, you can see that it's kind of associated with excessive or strong trained immunity responses. So the higher immune response we have, the higher chance that you have organ rejection, autoimmune diseases, and also cardiovascular complication. Whereas on the left side, where we have the defective trained immunity, then you have the lower immune responses to fight against infection and therefore leading to sepsis and sometimes also in cancer. So it's exciting, but we also need to see that we need per diseases, we need to see what is a specific role of this trained immunity into the disease pathogenesis and what are the impact of that in different cell type and how can we approach that for therapy. So in our lab, we focus on the endothelium part. And we show that in the past that endothelial cells, besides being the sentinel cell type, it also regulates inflammation by its interaction with immune cells. So we can see that indeed when we have infection, immune cell like leukocyte can recognize the pathogen, it secretes a lot of cytokine. And these cytokine, in fact, gonna activate endothelial cells to induce expression of adhesion molecules on the cell surface, but also to secrete a lot of cytokines like IL-6 and IL-8. And at the end, for the looping feedback, then it stimulates again the immune responses. So by blocking those cytokines, we have been shown that endothelial cells are actually interact with immune cells independent of what type of pathogen it is, mainly by interferon signal and TNF-alpha signals. So giving this kind of like interaction between immune cells and endothelial cells, we wanted to see how do these endothelial cells actually respond to the signals that are released from trained monocyte. And in more specific, we want to see what are the effects that happen to endothelial cells when we trigger them with TNF-alpha and interferon-gamma into the phenotypes of the cells. And we also further down characterize to see if actually endothelial cells can remember. And I hope after this I can convince you. And after this, you could have the answer. Do it remember? And the implication of endothelial cells' immune responses in different disease. So to answer the first question, how do endothelial cells respond to repetitive exposure of a cytokine like TNF-alpha? We use a simple model of endothelial cells, HUVEC, from 5 different individual donors. We sit them on well, and then we perform stimulation. So you can see here is the scheme of how we stimulated these cells, or the agenda of the cells. So the experiment lasted in 4 days. And the cells that we wanted to compare the responses of cells that have only 1 hit. You can see here is the 1 hit condition, and cells that have 2 hits of stimulation. So they have like first, on day 1, they have 1 hit, and then they was rested first, leave it rest, and then on the day 4, we stimulate them again. So we wanted to compare the responses between these 2 conditions. And besides that, we also have in mind to control for the time effect and the remnant effect of the first stimulation by having the 2 more control, in which we call control 1 or control 2. And you can see here on the graph below is the expression level of VCAM measured by flow cytometry. And if you look into the last two columns, we can appreciate that in cells that have 2 hits of stimulation, they have higher expression of VCAM compared to cells that have only 1 hit of stimulation. And we do it the same for E-selectin and ICAM, but we only see the significant induction in ICAM and not on E-selectin. So up to now, it shows that if we pre-expose the cell to a cytokine, then it actually can mount the higher expression level of adhesion molecules. But what does it actually mean with the interaction of that with the monocyte? So then for this, we welcome Helen with her expertise for making blood vessel on chip. And in this case, she used the PDMS chip with 6 microfluidic channels with the diameter around like 500 microns. And then with viscous finger patterning, we make the lumen relatively straight in the chip. And then we can see cells here. So you can see here is how endothelial cells lay confluently in the vessels. And you can also see on the standing picture on the right side for DAPI and also for the actin of the cells. So after sitting cells in the chip, we wanted to set up with the monocyte adhesion assay, in which we connected the chip with the pump, and on the other side you have the monocyte reservoir. And in this reservoir, the monocytes already stand with the dye, so that you can really easily to recognize them later in the fluorescent microscope. So we connected that, and then we performed the flow assay. Yeah, so for 10 minutes. And then you can really see that how monocytes are rushing through the vessels. And at the end, we stop the flow assays and then wash the lumen and then check under the microscope for the monocyte, the number of monocytes. And on the right graph here, you can see that in TNF-activated endothelial cells, we have way more amount of monocytes that are adhere compared to the non-treated or negative control of vessel lumen. So after setting up this assay, we come back to our funky experiment scheme, and then we, instead of using it on the chip, on the well now, we put them in the chip, and you can and then perform the adhesion assay. So you can see that if you look straight into the 1 hit versus 2 hit condition in GFP channel, we do see that in 2 hit stimulation, there are much more monocytes adhere to the lumen compared to the cells that have only 1 hit of TNF-alpha treatment. So this shows that if we pre-expose endothelial cells with TNF with different cytokines, they actually can remember and they can attract more monocytes than naive cells. So interesting. So that was really interesting, and we wanted to see more because we are immunogeneticists, what's happening underneath the cells. So then we will go straight ahead with the transcriptome analysis. And when we perform differential analysis between cells that have only 1 hit or 2 hit of stimulation, we can see that there are a number of genes, so 283 genes, that are differentially different between cells that have only 1 hit or 2 hit of stimulation. And interestingly, around these 283 genes, around more than 50% of them, 150, are actually still activated after the first peak of activation. So after the first hit, this one is still activated. And then another 133 genes, so around 40%, are uniquely activated by the second exposure to TNF-alpha. And if we perform pathway enrichment, we see that these genes involved in cell recruitment, in inflammasome activation, and also in translation of protein process. So we also wanted to see how these responses of the cells and how can we classify them into immune response clustering. So then until now, we can classify immune responses into three main groups. One is priming, training, or tolerance. So it really depends on how high is the cell response on the second insult or the second hit compared to the first hit. And in priming condition or training immunity, you can see that the amount of immune responses primed in the second insult is much higher than the first one. And the only difference is that in priming, the cell doesn't go back to baseline before the second hit. But in the training immunity, it has time to go down, all the cells become normal again. It's not so normal, but it looks normal on the surface. And then it's primed the second responses that is much higher. And in tolerance, you have less amounts of responses that you have on the second insult to the first insult. So coming back to our data, we actually have the transcriptome data that capture the status of the cells. So actually we have cells that have no exposure at all, cells that have one hit, so indicate 4 hours of stimulation, cells that have 3 days of resting, and then cells that have the second hit of stimulation. So then we use this transcriptome data to perform pseudo-time analysis. And then when we kind of map these genes, you can see that we can see 4 main clusters. So then most of the genes are involved in training or priming in cluster 1 and 3 and 4, whereas in cluster 2 and 3, you have genes that also show tolerance effect. So when we kind of enrich for what kind of pathway that these genes involved in, we do see clearly that in genes that are involved in training or prime, they involve in viral infection fighting, like COVID, influenza, or HPV, and for other cellular mechanisms like cell adhesion, for the interaction with the ECM, or the example of ribosome and the COVID fighting. So it's interesting for us. And then we also do the same for interferon gamma. So we do see less number of genes that are activated, but we do see common genes and unique genes that are activated by the second hit of exposing these cells to interferon gamma. And when we perform clustering, we do see that there are 2 clusters. So these genes actually can be trained or primed in cluster 2, in cluster 1, or tolerance or in cluster 2. And unlikely, different to TNF-alpha stimulation condition, where we compare them with different pathway, we do see that these genes are actually enriched for genes that are involved in autoimmune diseases, like type 1 diabetes, allograft, thyroid, or IBD inflammation. And for other cellular process, these genes involved in T-cell differentiation, in phagosome, or adhesion. And then another layer of this information, we wanted to see what is actually happening, like how these kind of stimulation shape the cell response the way it is. So we use ataxic method to really map the gene activity of these cells. So I'm not sure if you are familiar with the ataxic, but imagine that our genes are packed into chromatin, and there's some dense, but some also some open area. So then in the dense area, if genes are there, it's not activated. And if it's in the open area, then these genes are activated. So then with ataxic method, we can use transposase enzyme. So this enzyme can actually just randomly access to any open area of the chromatin. And you can imagine that if we just tag this TNF enzyme with some specific index of primer, and we put it into the chromatin, the nuclei of the cells, that it can cut any random open area of the chromatin. And then we can amplify this library and sequence them. And after that, you can just map back and to see okay, so this area is open, this area is closed. And we can compare between different conditions to see what actually happening at the chromatin level. So we did that, and then we saw that in TNF stimulation, so if you just stimulate the cell for the first hit of with TNF-alpha, you have around 5,400 peaks that are activated, and then most of them going down, and only 64 peaks remaining activated. And then when you activate them again on the second hit, you have around another 3,900 peaks that are activated. And if you wonder if they are shared or if they are unique for per stimulation, then we put it into the Venn diagram, and you see that indeed there's a large portion of them, the chromatin area, are shared, and 43% of this area is actually unique for the first stimulation, and 19% of these 3,900 peaks are unique for the second stimulation. And we'll also see the same for interferon gamma, so I just would leave it here in case you are interested. And then just to give the closure, so I would like to emphasize again about the importance of having this kind of data set to really see the role of endothelial cells and the microenvironment that these endothelial cells have in order to understand different kind of disease pathogenesis. So in this example, we would use sepsis, a blood infection, and we focus on the gene that called CETA genes. So this CETA genes is important for sepsis, and when it's regulated the level of HLA-DR genes, and with the lower level of HLA-DR, then you have higher mortality in sepsis. So if you look at this kind of data set, so in the first photo, we have this transcriptome data of endothelial cells, and then in the last photos is the ataxic or epigenomic data. And then if you look at only this area, so in green, you can see that on the upper line is the cells that have 1 hit of stimulation. Lower line are cells that have 2 hit of stimulation. And we do see that in cells that have 2 hit, you have higher expression of gene level at CETA. And we do see the same for ataxic data. So you have the more opening at the beginning of these CETA genes in a cell that have 2 hit of stimulation. And more interestingly, these CETA genes or these locus have been known to regulate, to have genetic variation that can impact sepsis mortality. And then in this data set, you can really see that this NIP is actually located in this area of the promoter. And with the continuously exposure of them to interferon gamma, you can see that this area of the chromatin is more open. So then it can impact really the impact of these genetic variation on sepsis itself. So just for that to really show you that the history of infection can actually sometimes override, sometimes can modulate the genetic impact on different genes that are important for diseases. And to conclude, so do endothelial cells remember? Yes, it does remember. And what does it remember? To the prominent cytokines of TNF-alpha and interferon gamma. And it does behave like a train, like tolerance, but also like prime phenotypes. And there's different mechanisms for TNF-alpha and interferon gamma. And I think like for future perspective, if we wanted to modeling some kind of disease model to study therapy for immune-related diseases, I think the history of exposure to certain cytokine or microenvironment should be considered carefully. And with that, I would like to thank you for your attention and acknowledge my team and the support that we have from UMC Groningen, from Radboud, and especially from the Netherlands Organ on Chip, the NOCHI, because there is actually where we meet, the biologists actually meet the engineer. And with the hands-on expertise from Helen, Nick, and Ling, and without you guys, I don't think this project can be done in the last 3 years. Thank you so much for your attention.
We have already here a question from the audience.
Hi, that was absolutely fascinating. So my lab years ago did a lot of work on antigen presentation by human endothelial cells, something that was unique to them, absent in mouse. I'm just wondering how this sort of plays into it. And so my specific question is, do you have any idea how long this memory lasts in the endothelial cells?
So then that is the millions of dollars question that we have in the field now. So from endothelial cells, we don't know specifically how long does it last, but like in the whole organism, their study have been shown that it can last for 1 year, 3 years, or to 5 years, depends on what you use to train them. So one of the evidence, kind of like agent that we use to induce training immunity is the BCG vaccine or the I forgot, just like the prime shot that you have before vaccination. So those one have like a really long effect, up to 5 years. Right. And there's a lot of study now in monocyte, for example, that they actually inject it in healthy individual, and they follow them for, in years. So then hopefully in like 5 years, when the cohort is like kind of like get to the time where they can analyze the result, we can answer your question.
Right. Yeah. So that's interesting. So you've shown it for HLA-DR, so class 2. So do you know if it also holds true for class 1? And does it hold true for things like TLR receptors, or the TLRs, things that might control innate immunity?
So then for this one, so then we have, up until now, I think a list of like 80 genetic genome-wide SNP that are located in different genes. And then one of the examples I show is the HLA-DR locus, because it's really show that there's a trained immunity happening there. But the interesting thing about this HLA-DR is that it helps with the expression of MHC class 2, which is normally expressed on immune cells. And in sentinel cells like endothelial cell or epithelial cells, we are not so sure. So some cells, they say, yes, it does express. Some cells, they say, no, it doesn't. So then, exactly. So then what is the implication of this increased expression of LADR and the zeta genes and interferon gamma and the downstream pathway into certain diseases? It's difficult to answer in this kind of like in vitro model. Yeah. And I think we still need to go to, a more, like the whole system to really answer that.
Okay, we have another question here in the back.
Hello, thank you very much for your talk. I was actually curious, so maybe I missed it, so I'm sorry in the case. Why TNF-alpha interferon gamma? So exactly why you focus on these two? Is because related to monocytes activation and so on? So I was curious if you are, I think you are planning to increase the level of cytokines. This is first question, and then I ask another one.
Yeah, thank you for the question. So then we select, so from the previous work, so we actually still wanted to study the interaction between monocyte, and in that case was BBMC with monocyte. So we stimulated BBMC with a lot of different pathogens, so from positive bacteria, gram negative, and fungi. And after that, we take the supernatant, add to endothelial cells, and then do all the blocking. And after that, we do see that despite of what we block, actually the endothelial cells only need the signal from TNF-alpha and interferons to drive the activation of endothelial cells. So those are the two main cytokines that endothelial cells are responding and connect to the monocyte based on what we saw from previous project. And then on the second perspective of this one, so we know that TNF-alpha and interferons are the two main cytokines that are really highly dominant in any kind of chronic inflammation or any kind of immune-related diseases. So it's either TNF or either interferons. So then because of that, we would like to go for that.
Yeah, that is amazing. I mean, it would make sense with macrophage, right? The differentiation, interferon gamma and TNF-alpha. I was curious, after the first wave, so you activate once, and then you wait a few days, you say 3 days from the second stimulation. So in between, you don't see any activation of cytokines by the endothelial cells. I was curious. Maybe I missed it, sorry.
Yeah, so then in between of so in that resting period, the cells are actually still activated actually. So I did have experiment where I let the cell to rest for 1 day, 2 days, or 5 days. And we see that after 1 day, a lot of genes are still activated, but after 2 days, most of them are going down. So at that time, we based on the secretion of IL-6, IL-8, and all the adhesion molecules, and we saw that, okay, after day 2, it goes to baseline. So that's where we decided to put the second stimulation. And I think that during that duration, the cell still keeps secreting, and then it's somehow either block or activating the cell. I don't know. Yeah.
Okay, we have one last question here.
Great talk. So I was just wondering some details, the procedure of your experiments. When you are treating the cells with the TNF-alpha or IFN gamma, what is the concentration you actually treat the cell with? Because sometimes if you give them really high concentration, the cell will just die.
So then we actually did the titration already with TNF-alpha, and then we use it from 1 to 100 nanogram per ml. And then we do see that with the 10 nanogram per ml, that's the concentration that we use for endothelial cells. And on HuVax, it works because the cell doesn't die, and it's still responding. And at 100 nanogram per ml, then the cell started to die faster. And there it's difficult to see. But I also did it on iPSC cells as well. And with this 10 nanogram of treatment to iPSC-derived endothelial cells, cell died. So then for iPSC, we still need to optimize the concentration of TNF and not to make it so cytotoxic for the cells.
So for both TNF-alpha and IFN gamma, you use 10 nanogram.
10 nanogram.
I see, okay.
Yeah.
All right, thanks.
All right, thank you, Kieu. And then let's also thank all our speakers and close the session. And yeah, sorry for the technical problems.